Hello everyone, I've got an A-level biology subject here. This is one of the sequence of videos that I'm going to make on biotechnology. Now it's quite in-depth, so what I'm going to do is break it into the small fragments in the way that I would normally teach a class about it. And in this particular video, we're looking at microorganisms and the standard growth curve. Now let's just give it some background and talk about how useful microbes are generally, and then I'll explain the setup that we've got on the screen. Now, anaerobic respiration of yeast produces ethanol in a process called fermentation. But moreover, the term fermentation may actually refer to the culturing of microorganisms both aerobically and anaerobically in fermentation tanks. So that's one key thing to just note there. Now, substances generated from the culture are siphoned off, so they're extracted off, and they're treated to produce a final useful product. Now, this is what's important. Precise growing conditions are needed, and they're controlled not only to maximise the yield, so how much you get in the end, but also to give predictable growth patterns. So we want to control things like temperature, the type and the time of the addition of the nutrient, the oxygen concentration, the pH. Now, in a closed culture, the conditions are fixed, resulting in these predictable growth patterns. No new materials are added and no waste products or organisms are removed. I just want to define the word culture because that's something I probably will be referring to quite a bit in this video. Now, culture is defined as a growth of microorganisms. Now, this may be a pure culture, where you've got a single species, or what's called a mixed culture, where you've got multiple species. Now, they can be cultured in liquids, such as nutrient broth, or even on a solid surface, such as nutrient agar gel. So there's a little bit of background info to fermentation tanks, what goes on in them, and the precise growing conditions needed to be able to culture certain bacteria in them. Now, what you have on the screen is what I give my students when I introduce the standard growth curve. So we've got here on the bottom of the axis, on the x-axis, the time after culture in hours, and up the side on the y, we've got the population or the cell number, or referring to bacterial cell number in culture. And I give them this, and I show them all of these boxes around the outside, and they have to draw an arrow from each box to what it's representing on this graph. So that's what we're going to do in turn. We're going to start from the bottom and work our way around the graph, start to finish. But except this time I'm going to show you all the answers. So at the very beginning we have what's called the lag phase. That's right at the start. Now in that lag phase we've got quite a few things going on. We've got this one that should be there, and we also have this one. So we've got three boxes there, all pointing to the same starting point, this lag phase. Now here, the bacteria are starting to just adjust to the new conditions. So they're active, but they are not fully reproducing. So we have quite a small population to begin with. It's taking time for the enzymes needed to be synthesized, to activate genes, for cellular expansion, for growth, and the uptake of water that's required by the bacteria. So you can see this would be our lag phase. We are lagging here. So our population, our culture of bacteria is growing quite slowly here. They're not actively reproducing, but the cells are active. Then you'll notice this sharp rise. We get this rapid increase in bacterial numbers. So we can now put in place this one at the bottom here, because what we've got is a rapid increase in bacterial numbers. The population size is actually doubling in each generation, as each individual now starts to have enough space and nutrients to reproduce. And obviously this part has a name, and this is known as the log or exponential phase. So this box here can so too join with that one there. So this is the log or exponential phase. We get exponential growth, rapid increase in bacterial numbers because they're thriving in that environment. They have the nutrients to be able to reproduce. Then you'll notice that all of a sudden the graph goes flat. On this standard growth curve we have this plateau. Now that, as the name would suggest, would be our stationary phase, things are stationary. You can see that it lasts quite a significant amount of time. On this graph, that's going from about 10 to 32 on the x-axis. So we're talking about 20 plus hours here in the stationary phase. Now, during that phase, what you can clearly see is that the rate of growth would be equal to that of death. That's why we've got that kind of steady population count at that point. Now here, this is where waste products and metabolites are starting to build up. Now, waste products can be toxic, and in large numbers can have an actual negative effect, which we'll come to. But at that point, we're starting to get the build-up of these waste products. Now, in an open system, this would be the carrying capacity of the environment. Carrying capacity, by that I mean the sort of maximum this can hold, for example. It's the maximum amount of population that can be sustained in that particular environment. And then we have this sharp decline. And we've only got a few boxes left to fill, so it's fairly straightforward from here. And I'm guessing you can think that from the name, this would be a decline or death phase. Now at that point, this is where our death rate is greater than the rate of generation. So the rate at which new bacteria are reproducing. And I said that the build-up of waste products and metabolites can have a toxic negative effect, and that's what's happened. So we've had this lag phase where things are slow to start, this log or exponential phase where you get a rapid increase of bacterial numbers, a stationary phase, and then finally a decline or death phase. And that, in biotechnology terms, is known as the standard growth curve. So what we're going to do is just look at two particular exam-based questions that I pose my students and talk through what kind of answers you could give for those two. So here are two quick questions. First one says, give 3 reasons why the death rate increases above the reproduction rate in the decline phase. So that's 3 specific reasons why we've got a greater level of death than reproduction. So let's think what answers we could have for that one. Well, in the first instance, we could have toxicity caused by the metabolites. Now, if we do have a large number of reproducing bacteria, there is likely to be competition, so we could put that as a second one. And because of competition, that would lead to really our third answer, which we're going to put next to that, because the two kind of go in tandem with one another. We would get a lack of resources. And by that, what I mean is our nutrient supply. If we've got significant competition for the nutrient supply from all this growing bacteria, clearly there's going to be a lack of it. So there won't be sufficient for them to thrive. So we've got competition, lack of resources, and also this build-up of toxic chemicals and waste products essentially in the tank also. All 3 could be a reason why the death rate is increasing above the reproduction rate in that decline phase. The second question here says, describe how adding nutrients to a growing culture of microorganisms might affect the population growth if they were added in A, the lag phase, and B, the stationary phase. Well, let's have a think. In the lag phase, if those cells aren't particularly reproducing at all, if they're active but not reproducing, then we're not likely to get a very big effect. It says, how might it affect the population growth? It wouldn't really affect that population massively, because in the lag phase, those cells aren't actively reproducing. In the stationary phase, however, so that's the lag phase, but in this stationary phase, we should get a significant growth, because now what we've done is provide a greater level of nutrient to that container, if you like. So now there's less competition for those limited resources, so more bacteria should, in theory, thrive. So there's 2 past exam questions that you may get asked on this. I hope this video helps. It's just a little bit about the standard growth curve and the use of microorganisms. So I'm going to pick up on the other areas of biotechnology in my other videos. Okay, I hope that helps.